For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 3, Episode 4 of Surfing the NASH Tsunami. Our wrap-up coverage of NASH-TAG 2022 starts now.
This week on Surfing the NASH Tsunami.
Having diverse stakeholders, the FDA, patient advocacy groups, therapeutics, diagnostics, all in the same room in one constructive conversation really will drive the field forward with innovations. Good science and good analytics leads to more questions and answers. And some of the key takeaways from the Fireside chat was if there's so many limitations with histology, this begs the question if we should stop correlating biomarkers with histology and how will this translate to different endpoints.
How do we think about this from the patient perspective? Are they going to really be told as patients that they need biopsies once, twice, maybe 3 times a year, depending on the therapeutic and what could come to market? That's not reality, right? I'm hopeful that we will have a strategy, depending on what type of analyses, what type of data we're going to be able to produce to make that happen. And it will take some thinking outside the box in order to make that happen.
When you talk about a screen fail rate dropping from 80% to 50%, if you flip those numbers, you're talking about an acceptance rate that goes from 20% to 50%, which means you're accepting 2.5 times as many patients. Then that makes the economics of trials, the ability to develop data quickly, the willingness to take risks, all that stuff changes because the cost of doing it changes.
Hope is not a strategy. That applies to drug development in NASH as well. Movement always starts with one person, and it's validated when the second person joins the movement. And here, what I've heard from you collectively say is multiple people join the movement Saturday night, including the FDA.
The first thing I would say is the FDA is slow to react to change. That is not always the case. And in fact, just given the events and what the FDA has approved in a matter of weeks, what's been put before them in the past year or so is a testament to the fact that the regulators can act in expedited fashion, particularly when there's high unmet need and when there is good data to support things.
Over 140 NASH and NAFLD stakeholders came to Park City, Utah, for the 6th annual NASH-TAG conference. This week, we have organized a panel of attendees to help process the conference's high points, key takeaways, and next steps. Join hepatology researcher and key opinion leader Dr. Stephen Harrison, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and today's guests, Novo Nordisk NASH Medical Director Amy Articolo, Terns Pharmaceuticals President and Chief Medical Officer Erin, Quirk, hepatology researcher and key opinion leader Professor Ian Rowe, and Age Diagnostics founder and CEO Rachel Zayas, as they review 2 amazing days in Utah. Today, on Surfing the NASH Tsunami.
So just for our audience to know, we're doing so many experiments at once today. For a podcast that never tries to do 2 new things at the same time, we're kind of breaking our record. For one thing, we have 7 people with us. We try never to have more than 6. 3 of them are newbies. We never try to have more than 1. We don't really have a focused topic. We don't have a paper we're talking about. We don't have in the narrower sense, a public event. We just have an amazing 2 days of meeting. In the pre-talk, Stephen shared with us his most interesting experience of the after meeting, which he might get to on the first go-round. What we're going to do is we're going to follow up on the weekend of NASH-TAG. If you're listening, and I know some of you did because the early numbers on the same day episodes are really good, really high. So we figured rather than talking more about everything we just talked about, particularly given Steve and I flew Louise yesterday and that Louise had some sleep to catch up on, that we were going to bring on friends who were friends from before or from the meeting and ask them to talk about how they experienced the meeting. And we will ask questions and occasionally share comments. So let me say hi to Louise and Stephen first. Louise, how are you doing this evening?
Absolutely fine. Thank you very much. And enjoying the new time slot. Nice to meet everybody.
For those who don't know, we've moved the recording sessions an hour and 45 minutes earlier to get them into Stephen's lunchtime and to make it more convenient for the Europeans. So what Louise is referring to is this isn't starting at 9 o'clock or some ridiculous hour for her. And then, Stephen, how are you?
I'm doing well, thank you. Just a little tired, not jet lagged, but just, I guess, mentally fatigued from the past 3.5 days, but doing well. Motrin's helping a little bit too, as Amy pointed out in the prelude.
Well, since nobody knows who Amy refers to exactly, we'll fix that first. One of our 3 friends who are new to the podcast is Amy Articolo from Novo Nordisk. Amy, how are you this afternoon?
I'm doing great. Thank you so much.
Well, we're delighted to have you with us. About the time this idea was coming together, I wound up bumping into Amy and a couple of her colleagues at breakfast on my way out yesterday. Turned around and said to Amy, so what are you doing at 1:30 this afternoon? And she said, gee, nothing. I shared this idea with Stephen, and I had a couple of ideas ideas in mind, but beyond that, I didn't. And so, Amy, I thought you'd be perfect. So tell everybody a little bit about yourself, what you do now, how you got here. And then, as I told you, one fact the audience wouldn't know about you if you didn't choose to tell them.
All right. Well, thank you so much. And just want to say, so glad we met officially over breakfast. And thank you so much for this opportunity. It's just a true pleasure working with all of you. I had shared with Roger that I have the app on my phone and listen to the Surfing the NASH Tsunami on a regular basis. So really, it's just such an honor and pleasure to be a part of this group. So it feels like full circle for me. Thank you so much again for the opportunity. And a little bit about me. So I'm currently the medical director for NASH with Novo Nordisk in medical affairs in the U.S. division. And I've been with the company for almost 4 years. I started out doing advocacy work with professional and patient organization, very passionate about patient advocacy. And the last 2+ years have been in medical affairs, working as a medical director. Prior to that, I am an OBGYN, and I was in clinical practice for many, many years. I've delivered probably over 10,000 babies, believe it or not, in my career. So I consider myself very blessed of all of the career opportunities I've had. And what got me into metabolic diseases was approximately 8 years ago, maybe almost 10 years ago now, as time flies, as we were transitioning at the hospital system from being traditional labor and delivery units into more sophisticated and collaborative maternal child health units. We were looking at what are areas of improvement that we could do for maternal health. And of course, what bubbled up quite a bit was metabolic disease. I can remember when we called metabolic syndrome X, and we didn't do much about it in the OBGYN sphere because we didn't know what to do. So what doctors do when they're not sure about things, they look for education. And so I went to every educational congress, scientific event that I could find. And that's how I met a lot of my Novo Nordisk colleagues. We got to know each other, and the rest is history. Here I am, and it's a pleasure to be a part of that organization and a pleasure to be here. So the one thing that people wouldn't know about me, so I think we already covered, I'm a big believer of Motrin, so I always carry that in my bag, but now everybody knows that, so that doesn't count. The other thing would be, I don't even think my husband knows, okay, so you guys are the first, is that I really don't have confidence in my singing ability or capabilities. They're pretty much zero. Like I even sing Happy Birthday off key. Ask my children, they'll tell you that. But I love to sing rock and roll in the shower. So that's the one thing that I don't even think he knows, because I'm so embarrassed by my voice that I pretty much ensure that it only happens when I'm alone. So that's my interesting fact.
So I have to tell you, you actually had me at the 10,000 babies, but that's a real bonus, okay? Okay, and welcome. Delighted to have you.
Oh, thank you.
If I go around the screen, next I get to Rachel. Rachel Zayas is the CEO and co-founder of Diagnostics in Washington, D.C. She's been a listener. She wrote me a note a while ago, and when I said to Stephen, I think we might invite Rachel on, he said, oh, Rachel came to NASH-TAG because of me. So, Rachel, we're fighting for ownership of you, but why don't you go ahead and a little bit about how you got to what you're doing, and then your one fun fact.
Yeah, so I grew up in Cambridge, Mass, and I was down the street from Harvard and MIT, and by design, I went into genetics. I was down the street from all of this wonderful research. I got a degree in genetics, and then I worked at MIT for a number of years. While I was there, we were developing a gene-sensing therapeutic for different types of cancers, and I had a lot of fun developing these nanoparticles. And at that time, I realized that the tools that we were using in research were not being well translated into the clinic. At that time, I went back to grad school, and I got a dual degree in genetics as well as an MBA. I knew I wanted to start a business, but I had never taken a business class prior to grad school. So I spent a couple years developing that foundation, and I launched my first biotech company in 2015 when I was just in my early 20s, and that failed, and it failed fast. And you learn so much from your failures. So we took the knowledge from that company, and I started Diagnostics in 2018. We're a team of geneticists and several hepatologists, and we're developing non-invasive tools for fatty liver and fibrosis. We are taking a really underexplored aspect of the human genome called epigenetics, which has been falsely called junk DNA for the last 50 years. And lo and behold, that nomenclature has really come back to bite geneticists in the backside, because that junk DNA per se really orchestrates everything from disease to longevity to health and wellness. So we're looking at these non-coding regions, and that's a little bit about me. One fun fact about me that nobody knows is that I have written 2 fiction novels. They're sci-fi novels about searching for meaning, and it's really just my bread and butter. At the end of the day, I usually write for 30 or 45 minutes. I find it fun and creative, and Those books should be, the first of the series should be published by the end of the year. So that's one fun fact. And I'm really happy to be here. Thank you so much, Stephen and Roger, for the opportunity.
So Rachel, that means we're committed to bring you back at least once more after you publish so we can do a meet the author thing. You know, we're going to tie that into NASH somehow, but we'll get there. Okay, our third new person who's been with us before is Erin Quirk, the President and Chief Medical Officer of Terns Pharmaceuticals. And Erin, hi, welcome.
So thanks, Roger. It's so great to be on the podcast after listening to it since its inception from the very beginning. We always get together at my company and listen to the podcast every week and have a discussion afterward most of the time. So I'm Erin Quirk. I'm an internist and infectious disease specialist by training, and I am currently the President, Chief Medical Officer, and Head of Research and Development at Terns Pharmaceuticals. I had a brief stint in academics at Washington University in St. Louis, where I trained in infectious disease and internal medicine, and then started my industry career in 2005 when I went to Merck to initially work on HIV vaccine development, but then transitioned over into hepatitis C small molecule development. So I worked in drug development, liver diseases in the past. Moved to the San Francisco Bay Area, where I still live, in 2010 when I went to work for Gilead, where me and my teams got a number of different drugs approved for the treatment of HIV infection. And really, I think one of the major innovations we brought to the field at that time was the co-development of experimental combination medications. So when I first heard about Terns, I was excited to go there in January of 2019 because of the ability to work on combination drug development, which we think is going to be really applicable to the treatment of NASH overall. So that's me and my background. Like Amy, I'm a bad singer, but what you wouldn't know about me, I think people know I'm a bad singer, but what they wouldn't know about me is that I am excellent at step aerobics. Me and the other dozen and a half or so with our exercise regimen stuck in the 1990s to get together on Thursday and Saturday mornings at my club, do really complicated choreography, and it's super fun. It's my favorite workout. So that's me.
Well, that's super. Now, audience knows the names of 6 or 7 people who are with us. And the 7th is our good friend Ian Rowe, who's not been here for a while. But Ian, how are you this evening?
Yeah, I'm very good, Roger. I'm also enjoying the new time. Slightly more sociable than it was when the meeting finished on Saturday night here.
Yes, and Ian and Louise both stayed awake all the way through the chat, which in London and Leeds would put you 3 or 4 in the morning local time. Yeah, I think, Amy, I did make the comment on yesterday's podcast, which is only dropped in the last few hours, that we're going to have to name some kind of award for Louise for doing that 2 nights in a row for this meeting. We haven't decided what it'll be yet. Might have something to do with a lot of caffeine. Ian, since the last time you and I have spoken, your football team picked up a promising beginner and lost a troubling match. Stephen put one of your papers up in the middle of the Fireside chat as an example of things that we're learning. And I said, gee, how cool is that? So how are you today?
I'm very well, Roger. I think that the encapsulation of the ups and downs of football is a bit like the ups and downs of academics. Always great to see papers that you've written shown in meetings and influencing the direction of travel. But inevitably, almost the first official email I had of the year was of a paper being rejected. So it's the topsy-turvy yin and yang of the life of an academic.
Well, I think that makes sense. And that might be a real good thing to keep in mind as you follow Newcastle over the next year as they try to get their legs in their budget aligned. I think that's a good thing. So why don't we just dive in? And for those of you who, all you folks listen, so you probably hear this, but one of my favorite rules goes brave one first. So brave one first. What was the one thing about NASH-TAG that most surprised you? Stephen, Louise, and I are off the table for answering these things. It's the 4 of you. One thing that surprised you the most about NASH-TAG?
So I'll be the brave one first. I would say the energy was what really stood out for me. I mean, I've been through countless congresses and meetings, and you definitely get a sense of the fatigue over time. And for this event, the passion just shone through from the participants, the faculty, and the attendees. I mean, it was really something that stood out to me about how passionate everyone was and how empowered they were, the curbside consult, if you will, that were happening in the rooms outside of the meeting, the attentiveness of all of the attendees, the true energy really shown to me, and the record-breaking attendance, just a testament to all of you, and fantastic job and fantastic meeting. Really, our team that attended were just thrilled to be there, thrilled to participate, and really were just impressed with the caliber and the true energy of everyone that attended the meeting.
And we're so glad you showed up. And it took 5 years, but you got here as quick as you could, and you guys were a tremendous participant. So thank you for that.
Amen. Anybody want to either amplify on that or go in a different direction about what really struck and surprised you about the meeting?
Well, maybe I'll come in there, Roger, only to say that the final New Year's resolution that I'll make this year is one for next year, and that is to actually get to NASH-TAG in person. I thought that the virtual experience was good, but judging by the energy that you could feel during the talks, and particularly through the podcasts daily afterwards, it was obviously the place to be last weekend, and I'll do my best to be there next year.
And we'll save a spot for you, Ian, for sure.
Yeah. And Stephen, I was thinking that the energy was one of the things that lifted Ian and Louise and other people I know in Europe up to stay all the way through that second evening, because that was late for them and they were great with it. Rachel, go ahead.
I'll say two things here. I'm going to echo what was previously said with the energy and excitement and the diligence in the room. It really brought me back to my roots and really why I went into biotech in the first place. And you could just tell how hungry everybody was in the room and passionate. And I think that really spilled over, and it really has set the stage for such an incredible year for NASH and fibrosis. So that was the first and foremost thing that stuck out. Then I'm going to touch base on the elephant in the room. I'm going to go right into this. I was very surprised that there was such a substantial divergence in pathologists reading for hepatocyte ballooning. It seems very subjective. It seems like this has been something that I was surprised hasn't been under a microscope sooner. So that was the big aha for me. And I was very interested to see how this subjective perspective will have implications for biopsies in the future, and also lessons learned from the way that they're read. So what other ways have physicians failed to really classify these stages, and how can we take this lesson and translate that into other more effective tools coming forward? So that was a very interesting point for me.
Yeah, Rachel, just to follow up on that, one of the things we talked about in our podcast following the meeting was what one particular slide stood out for you or left an indelible impression on your memory. And of course, that one ballooned hepatocyte out of 1,100 that all 9 pathologists agreed on was one of those. And in that same vein, a slide from Naim Al-Khoury on the chimera, linking that to the ballooned hepatocyte was another one. And then finally, you mentioned the elephant in the room. I thought Sam Klein's last slide of his presentation was a bit poetic, but it drove the point home that it's a good time to be obese and have fatty liver because of all the drugs that are coming out that could potentially modulate weight loss. But you walk away from such a robust meeting like that and you try to remember key salient features or points that can help you in your own clinical practice. And to me, I always like to think about what slide could I throw in the back of my head and remember. And there were many that stood out, but you highlighted a couple for me there.
Yeah, and Rachel, I have to commend you also showing your writer's chops really quickly, describing the slide about the ballooning as something that we put under the microscope is really metaphorically, I think, strong. If you didn't intend to do that, you're better than you think. And if you did intend to do that, that was really good. Stephen, the slide you didn't mention that was the one that jumped out at me was Michael Charlton doing the thing about combination therapies and putting the hearts on all the good combinations, lest anybody forget what you're supposed to visualize. So I thought that was another really exceptional slide. We'll come back to
and maybe metaphorical in the sense that we don't talk about cardiovascular disease endpoints as much as we should relative to NASH.
Yeah, there you go. That would be also right. So, Erin, let's come to you then. Same question. We've taken away a few topics, but go ahead.
No, that's okay. I still have an original one. So I, for one, was not surprised about that energy because I knew this crowd and the energy that they bring to everything. And having listened to virtual NASH-TAGs in the past, I was only pleased to see the energy still emanating, which I felt virtually as attending the conference all the way through. I guess what I was surprised about was also a point that Mike Charlton made, and that so few NASH clinical studies are capturing lifestyle of the patients that are involved. I think he said he's only aware of one industry-sponsored study where food content is being tracked. And that was amazing to me because, Louise, I'm very much on your side of these things. These drugs will need to be used in combination with lifestyle modifications, as a supplement to lifestyle modifications in order for them to be most effective. And so if sponsors really aren't paying attention to diet quality, diet amounts of food, and then exercise as well, because we know that can have an impact on liver health as well, then no wonder we're seeing divergent and variant placebo effects across these studies. So I think it's something that all of us who are in the clinical trial design space need to pay attention to moving forward.
Erin, the reason that surprised me so much is that when Mike does his year in review, he sums up with something very visual and very memorable, like the way he used the Dalai Lama this weekend. 2 years ago, he closed by talking about Babe Ruth, the baseball player, and he used that as a metaphor to talk about the idea that the easiest way we can improve our clinical trials would be to manage diet. Literally, he said the one thing we can have the greatest impact on quickly is manage diet. And it was so obvious and so smart the way you put it together, because it's Michael we're talking about, and so self-evident that I had expected that people would have made progress on it. I don't look at clinical trials for a living, but I would have expected progress. So when that came up over the weekend, I was tremendously surprised that not more people had heard it.
We all stratify for vitamin E use to make sure that drugs that could potentially impact NASH are at constant levels, coming into the study, but we're not paying any attention to diet and exercise and capturing that generally. That's something that really could be an important addition to the field if we were to start measuring it, even in a small way in our clinical trials.
Quentin and Steve did comment that litmus collecting dietary information, and I agreed. And I texted Roger during that session to say I was struck by Mike's discussion on diet and lifestyle, because there are 6 types of different vegetarians. But if you're a vegan, if you're a Vovo-vegan, these all matter. One of my take-homes was the gut dysbiosis session, whereby we can look at things and look at how we can block it, how we can do it. I made the analogy of if we add vitamin A and C to food, we can increase the iron intake, but we can block iron intake by adding black tea, up to 70% of the iron. Now that's helpful for hemochromatosis. Unless we look at nutritional intake, and particularly when we look at weight loss, which I'm sure we'll come on to, in 12 weeks in some of these medications, what's the nutrient changes that we're driving? And these micronutrients through people that can have such an effect. In 12 weeks, that's a significant shift. But I've not seen any data of what we're doing to people nutritionally following these drugs and weight loss.
Louise, not the first. As Erin points out, not the first time you've ever talked about this. And in fact, I thought it was one of the major themes of the weekend is how little care we're paying to understand everything about the patient that isn't about their drugs.
One of the things we're learning with clinical trial development is there are ways we can begin to capture that as well. Online, virtual apps that can break that. And Michael's done a very good job of explaining some of those opportunities that exist as well. So, yeah, I agree. I would like to see a more granular tabulation of nutrient intake in patients in clinical trials to see how much that modifies over time and if that correlates at all with some of the changes we're seeing on the back end in our NITs or histopathology.
People are nodding. Anybody want to add anything to that particular thought, or we want to go on to the next topic?
I'll just add one more thing to that. When we go to market with a drug, or when we as healthcare purchasers buy medication, we need to know that it's the medication that's working, and that's what we're paying for. If 70% of the response of somebody's change of NASH or NAFLD is down to diet, and we pay for a medication that's highly expensive, then we need to be getting, as Stephen, can I get the phrase, the squeeze is worth the juice? Or the juice is worth the squeeze?
Juice is worth the squeeze, or the view is worth the climb. Yeah.
So we do need to know that when we're buying a medication to help patients and anybody with the problem, that we are buying the medication. And we've maximized everything else.
It's a great point, Louise. Let me give you the flip side of that one, right? Which is that we also know that drugs perform better in clinical trials than they do in practice. And we never know exactly why, but we always blame whatever. It seems that the ability to align diet to drug will, A, make the purchase more worthwhile, and B, increase the probability that you're going to get what you were looking for in the first place. And I'm mindful of the vegetarian class you've not mentioned yet, the famous pastatarians. People think if they eat pasta every day, then it's a healthy diet because there's no meat in it. But yeah, I take all that. All right, next question. We've talked about a few things that came up in the meeting outside of the Fireside chats that were really striking. Was there a particular presentation that wasn't in the last 2 hours or the first half hour where the entire presentation just kind of shook you, made you think differently, or made you say, boy, I'm glad someone's talking about that because that's way overdue? Either one.
I'll be the brave one here. So one talk that really stood out for me was the talk on single cell transcriptomics. I think it's multifaceted. That was an excellent talk, and I really foresee a future where single cell transcriptomics will identify drivers, and that will aid in more effective therapeutics. On the flip side with that, we need to be careful with single cell transcriptomics because it might, as a diagnostic tool, because that might lead to the same challenges right now with histopathological assessments, because there might be sampling error along with this. So I was really interested in this as a therapeutic, but I was cautionary in the use of single cell transcriptomics as a diagnostic. So really got my mind thinking. I'm not sure if anybody else had the same insights from this talk or wants to challenge me on this.
Well, I'll just add my 2 cents into this. I first kind of became interested in single cell transcriptomics when I went to Paris NASH, and I heard a very similar lecture. And then right after that, I heard Scott Friedman get up and have an elegant discussion on stellate cell biology. And you heard a little bit of that come back around in Scott's Q&A session where somebody asked him about senescence. And what we learned is that as stellate cells age, they don't go quietly into the night. It's not the golden years for stellate cells. They're bad actors. And in fact, we know that because when you apply CAR T therapy and you target senescent stellate cells and you knock them out, you get a dramatic reversal of fibrosis and serum liver chemistry tests, at least in an animal model. So there is a whole world here that needs to be explored. And I wonder, we talk about fibrosis in NASH patients, and I believe it was the Sinacrivarok Centaur trial where we did 3 biopsies over a 2-year period of time. We had different cohorts, including a placebo cohort that was re-stratified. And so we actually had a placebo cohort for 2 years and then one for 1 year. And we were able to show that fibrosis was very dynamic. In fact, it improved in 20% of people. It worsened in 20%, stayed the same at around 60%. And we thought that that was potentially just a natural history of this disease. Well, it turns out, well, maybe it's like the old joke I used to say on the podcast before. How many psychiatrists does it take to change a light bulb? 1, but the light bulb has to want to change. And it's the same thing with stellate cells. You could have a drug that could be very impactful on stellate cell biology, but if through epigenetics or some other mechanism, the stellate cell doesn't want to change, despite the fact there's a medication knocking on the door wanting it to change, it won't change. And so we could be swimming upstream with a lot of our therapies, and maybe in part that's why we haven't seen a pure antifibrotic actually be that effective.
Stephen, I think there's a lot to be said for that, and that whole session was excellent. You can never help but be impressed by talks that Scott Friedman gives. They're always fantastic. And I thought that the point that he made about unmet needs in fibrosis was when we think about antifibrotics, we're thinking about drugs that are going to block the progression of disease. And what he was really talking about was trying to understand resolution of fibrosis more clearly so that you could target the other side of the balance. So that once you've switched off injury, or maybe even without switching off injury, then you can promote the resolution of the scar, allow the fibrosis to regress, and for the patient to have a better outcome. I guess putting those two things together, the team in Edinburgh are running studies looking at macrophage-based cell therapies to try and promote scar resolution, really as proof of concept that that can be done. There's lots to be seen through the approaches complementarily, looking at both progression and regression.
Yeah, and I think that your point is incredibly well taken. In our drug development strategies, we have tended to say, we'll knock out the upstream drivers of stellate cell activation, and vicariously we'll wait for the liver to begin to heal, rather than targeting therapies that could actually promote wound healing in addition.
And to that end, I'd love to add, I really appreciate those talks because I'm a big fan of let's get to the core of the problem, right? You know, what are we really looking to drive and really solve for? And so that for me, I felt like it was the aha moment of the talk that Michael Charlton gave regarding combination trials and his hearts along, you know, what he would like to see, because it resonated quite a bit with me, not only as an individual, but also, you know, from a company that's striving to look at combinations, but also to hear Scott talk about that and the upstream effect. And so it makes you pause and go, okay, so what are we really trying to do? You know, what is the greatest impact for the greatest good in a very patient-centric approach, thinking what is the patient going to be willing to take to improve how they function, how they feel, and what can we as collectively do to really get us from point A to point B so that we're able to put that therapeutic and that potential combination therapeutic in the patient's hands.
Yeah, I think that's great. When I told you at the beginning about the things that we never do, I think this may be the first time in the history of this podcast we've ever had 2 people from drug companies on at the same time. And I mention that because one of the things I commented on both days was one of the things that kind of an undertone on the meeting, I thought, was the challenges caused by a system in which so much of the drug development is done by small companies. You know, Michael talked about that as a phenomenon right at the beginning. I thought Vincent Wong's exceptionally skillful presentation of the aldafermin data touched in several different ways on the idea that if you had more money, if this was developed by a company that had more money and greater latitude, it might have gone in a different direction. And we see in a whole bunch of ways as we go back and look at the development process that people have to make compromises with the ideal in designing their trials because they can't afford not to. And then as a result, bad things happen and we get stuck. I made the comment to Amy at breakfast yesterday morning that I thought that Novo was clearly an exception, if not the exception, to big pharma really being invested in this right now. I will note that. So, Erin, I'm kind of looking at you. How firmly did you feel that issue as you listened to the meeting, as compared to how keenly do you feel it every day, which I'd be happy for you to go into also?
I think this is one of my pleasant takeaways at this meeting, is that small companies get to be alongside large companies and present our data and be a part of the conversation, which I think is nice. Having worked at large companies in the past, it's had the advantage, I think, of having experiences in working for a big pharma and then also for a small company as well. In some ways, necessity is the mother of invention, they say, right? So the limited resources we have at small companies oftentimes does make us be a little bit more focused. In many ways, that can be a good thing, actually, in terms of what's going to be most important for patients or what's most important for any particular clinical study or what's most important for a certain pipeline. We are at a shift now. I would say if you looked at 3 years ago, there was a lot of activity from large pharma that really were driving the clinical trial landscape overall. So things have shifted a little bit. But in many ways, I think that might be a good thing for the field because most of the innovations, most of the new drugs do come out of smaller organizations in the end. So this meeting in particular was a really nice way for us kind of all to come together from different walks of life, talk about our data and think about what makes most sense, and then kind of go back to our day jobs and put those things into practice.
With the time we've got left, why don't we switch to Fireside Chat? Because there are at least 7 or 8 facets of that I think are probably worth talking about. I sent out notes for this meeting and I had a question in it, and I'm just wondering, on a 1 to 6 scale, where 1 is completely disheartened and 6 is over the moon, how did you feel at the end of that session, the Fireside Chat session? Brave one, go first.
I'd say I'm a solid 5 coming out of that session, Roger. The main reason is by the very virtue that it happened. That we were able to have representatives from a major regulatory authority alongside academic thought leaders and pharmaceutical developer representatives and diagnostic representative developers and sharing our thoughts and ideas. So that's something that you don't often see happen, in a public setting, I would say. And number 2 is I do think that there were some real issues, that were discussed and some opportunities, some doorways to opportunities that were potentially opened as a result. For that reason, I'm on the optimistic end of the spectrum and pretty strongly so.
Okay, next answer.
So Roger, I would agree with everything that Erin said, and I would say I'm a solid 5. What I really loved about this is the amazing forum that everyone came together. It is not easy to get all of those stakeholders in one place to share not only what was, what is, and what the future could look like. And kudos to everyone that put it together. Stephen, you did a fantastic job at really setting the stage in that initial presentation and then bringing everybody together to have a discussion. So I really applaud how it happened and the way it happened, and really was optimistic at the future of what things could look like. What I hope to continue to see and what I heard was the collective we, all of us, have to come together and work collectively towards this goal and ambition to really bring therapeutics in the hands of patients so that their lives are improved. That's what I heard, right? What I look forward to seeing and hope to see, as I'm a big fan of, okay, why does it matter? What is this going to do for the patients, and how are we going to do it? What is the next best step? Is it retrospective analyses? Is it more of a prospective design? That's what I would really love to see happen as a next step, and hope to see happen as the collective we.
Just parking this note after we get to answer Stephen, 2 days in a row we did not talk about NAIL-NIT. It seems to me that part of the way to respond to what Amy just said is for you to talk about that a little bit. But
I was hoping that would be a segue to do that.
Yeah, okay. I want to hear from Rachel and from Ian first, and then I'm going to ask Stephen to do exactly that.
So I'm going to go with 5 cubed. I'm going to say that the Fireside Chat was a 5 for me as well. I think that having diverse stakeholders, the FDA, patient advocacy groups, therapeutics, diagnostics, all in the same room in one constructive conversation really will drive the field forward with innovation. So it was exciting to see that conversation. Good science and good analytics leads to more questions and answers. And some of the key takeaways for me from the Fireside Chat was, if there's so many limitations with histology, this begs the question if we should stop correlating biomarkers with histology, and how will this translate to different endpoints. So I think that there's a lot of questions to be had here, and this really has set the stage for constructive conversations over the next 12 months. So solid 5 for me.
Okay, Ian, you're left.
We've talked a lot about this topic, and I very much enjoyed the conversations, and I thought that, there were some clear steps forward. There's still a very long way to go. The chats were a good start. again. So 6 is over the moon, then I was probably closer to 4, maybe even a little bit less. And one of the most important questions that was asked by the FDA is, what do you do when you do a biopsy for cirrhosis in the clinic? Well, we don't, and haven't for 5 years, probably 10 years, because we know that the non-invasive tests are so good. The issue is around not about cross-sectional analyses about how good they are, because we know that and we use them all of the time. The issue is about what happens prospectively. And because the natural history of liver disease is long, Vlad talked about natural history of cirrhosis and early cirrhosis of being 5 years, 10 years. It means that the time to accumulate sufficient data about a non-invasive test of whatever type accumulated prospectively and that shows a reduction as a surrogate in a trial means that we are still a very long way away. And it seems relatively unlikely, at least in the way that I heard what was said, that the FDA will move from biopsy to a non-invasive test based on cross-sectional data, because it might, the leap of faith that Michael suggested to them, it's exactly that. We don't know whether changes in non-invasive tests and what magnitude of changes in non-invasive tests are linked to an improvement in outcome, same as we didn't know for biopsy. So it may be that we'd end up jumping out of the frying pan and into the fire. The FDA have got an extremely difficult job to try and balance that. We talked a little bit about favourite slides, so I'm going to tell you my favourite slide, and that was from Dr. Shapiro. NASH plus cash equals ASH, and the balance here is about whose cash it is. I see patients all the time with end-stage liver disease. I work in a transplant centre and look after patients who've got HCC. We need treatments that are effective at reducing the rate of disease progression. But at the moment, we're talking about investor cash, we're talking about company cash, we're talking about patient risk. If we take risks with non-invasive tests by changing, we might delay licensing of effective drugs, perhaps. But what we might also do is transfer that cash that's turned into ash from the private sector into the sector for caring for patients. The most important thing to me is that we get drugs to market that are effective in doing what we want them to do, preventing progression of liver disease. We have to see some of the data from the outcome studies before we can have any confidence about the impact of biopsy or non-invasive tests.
Yeah, and I think that's absolutely true. And it's interesting to hear the 4 viewpoints, and whether it's a 4 or a 5. Either way, I think everybody walked away with a positive viewpoint. What I'll say is in the military, hope is not a strategy. And I think that applies to drug development in NASH as well. But the other thing I would say is a movement always starts with one person, and it's validated when the second person joins the movement. And here, what I've heard from you collectively say is multiple people join the movement Saturday night, including the FDA. And understand that the FDA has to be very calculated in the response that they provide. And you could see that in Dr. Turner's as you looked at Dr. Turner, you could see the wheels were turning in his head, and he was very calculated in what he would say to Erin and what he would say to everybody else. But you could read between the lines a bit and understand that he is battling bureaucracy, and he is battling an institution that is slow to adapt to change. And he wants that firepower, that ammunition to take forward so that we can help drive change. What I don't want to abandon in the short term is the fact that we are beholden to some sort of histopathology, in the sense that we have trials that are currently enrolling. We have trials that are currently screening patients. And while I think it's fantastic that we can jump to a discussion about NITs as an endpoint, we also have to be cognizant of the fact that today, right now, we are still screening people using histopathology. And so we also need to focus efforts on how we can improve that particular aspect of it while we look forward to the day we replace that with an NIT. We have a twofold job to do. We have to get a clear understanding of how we can use histopathology in the short term. And I thought Dr. Turner was clear when he said he is willing to listen to alternative endpoints relative to histopathology. And we need to work on that immediately. Number 2, bring forth the data that we need to generate to show him and his colleagues at the agency, particularly the statisticians, what it will take to move away from histopathology as a surrogate and then as a full endpoint. So to do that, one of the things that I've been able to sit back and think about a little bit is what's the role of the consortia that have come together to date to try to address these issues. And we have Litmus, and we have Nimble, and we have Goldmine that Rohit Lumba has initiated. All of those data sets are going to be very helpful in trying to address the NITs from 3 different contexts of use: the diagnosis of the at-risk NASH patient, monitoring for therapeutic efficacy, and long-term outcomes. But there's still a gap, and that gap is twofold. One, nobody has broken down the stovepipes of what each pharma company is doing individually to bring them together in an agnostic biorepository to ask the right questions that the agency and that everybody else is looking for. We can do that, and that's why we started NAIL-NIT. So that's the retrospective part of that. But furthermore, there's a way that we can augment and bridge the gaps between these other consortia. And so we want to do a further progressive or even just a prospective trial where we enroll many, many patients collectively in a real world setting, not in an academic setting, but in a situation where it's likely these drugs are going to be used in the real world. So generating that data prospectively in a community setting, not using liver biopsy, but driving the whole paradigm from an NIT perspective will be very helpful as well. And so those are the 2 aims of NAIL-NIT that were launched at NASH-TAG this year. But I would say the third component that I'm walking away from NASH-TAG thinking, and it's got me working on it today even, is we need to put out a paper describing exactly what the issues are and how far we've come with NITs and what that data could mean, and provide some degree of assessment on where we stand today and what we still need to do to get us across the finish line. We need that paper. And then the second one is, why can't we take the guidance document? The FDA was very clear. Dr. Turner was very clear. It's still a draft. Why is it a draft? Because there's some issues that haven't been resolved. Can we potentially use NASH-TAG and the momentum that's been generated to maybe add color to that guidance now that hasn't been done yet? So I think I'm walking away with a mandate that the FDA is receptive to change. We have generated more data than people individually, even myself, have realized that we had. We just need to pull that data together and see where we are. And maybe we don't get to an NIT this year, even in cirrhosis, but I do think we can begin to change the paradigm in a much faster way than we previously thought we could do.
Thanks, Stephen. There was a lot there, and I wholeheartedly agree with most of what you said. I guess the first thing I would say is the FDA is slow to react to change. That is not always the case. And in fact, just given the events and what the FDA has approved in a matter of weeks, what's been put before them in the past year or so is a testament to the fact that the regulators can act in expedited fashion, particularly when there's high unmet need and when there is good data to support things. But that's the key. And that's what you were saying at the end, which with I fully agree, is there needs to be data. I'd also like to react to what Ian said, and that we have a long way to go. And there probably is a long way to go until we can fully get rid of liver biopsies in clinical studies and rely solely on non-invasive tests rather than histological tests or clinical outcomes to get drugs approved in NASH. But that doesn't mean it's impossible. And in fact, the journeys of these thousands of miles start with these first few little steps. So number one, I'll say again, I've been in many different settings with many different representatives from many different regulatory authorities, and they generally don't say a whole lot more than what has been publicly issued in their written statements and their written guidances. So I've heard some people say that they were disappointed that there weren't more definitive statements made by the regulators on the workshop. But actually, I agree with you. I think there was an openness, a real openness to be wanting to be part of the discussion. And I think that's great. Involving the community more in that discussion is critical because the regulators are there to serve the public health, right? And so I think if we have these workshops in the future at NASH-TAG, I would love to see community and patient representatives on the panels, not just in the audience making commentary at the microphone, but on the panels themselves, because it's a critical point. It's the patient's disease after all. What we can start doing though is generating the data sets. And let's go back to something that Dr. Turner mentioned several times during the panel, and that is the example set in the HIV field in the 1990s. And that's something I know quite a bit about, having worked in that space for many years and being an HIV specialist in a former life. So, the first drug for HIV was zidovudine. The papers were published in the late 80s, and I think the drug was approved in 1990. And then there were a whole bunch of other drugs that were approved in the same class, but they stopped working because the virus got resistant. And it wasn't until the onset of the protease inhibitors and we figured out it's all about the virus, stupid, and we started combining multiple mechanisms that we were able to get people to live longer in the mid-90s. And then it was in the late 90s that HIV viral load was accepted. So think about that. You have to have effective interventions that move the biomarker so that that change in biomarker can be associated with the clinical benefit, which in this case was surviving, right? Or lack of progression to opportunistic infection, so late-stage disease. So we need to start having effective drugs out there for NASH, is the bottom line. We need to have effective biomarkers for how to measure them, and I think we're getting there. And then it's going to take time to accumulate the data to show that that translates to a clinical benefit. So yes, Ian, I do think it will be some time for those data sets to mature, but if we can start working together and pooling the data that we have, writing these consensus papers, and maybe even changing practice guidelines about how biopsy is used in practice or how the NITs are used even in diagnosis, that can be one of those important very first steps where we can start putting fewer needles in fewer patients' livers.
Erin, that's really helped me understand a few issues that Dr. Turner talked about. And I guess a couple of big issues that we face. One is that the natural history is very long. And unfortunately for those patients with HIV, AIDS in the late 80s and the early 90s, that wasn't the case, meaning that the time between diagnosis and severe illness was often short, and so that you could see an improvement in prognosis quite quickly. And in the context of cirrhosis, I was reflecting on the guidance from the FDA that they were all to be endpoint-driven studies. And I'm now wondering a bit, based on what Vlad said in the meeting, whether that guidance is, from a company perspective, a disincentive to doing studies in the patients who've got the greatest need, because where there's no potential for early approval, you're embarking on a study of uncertain duration, and where there's very little evidence about whether you can really prevent people progressing to the point of decompensation. And that makes me wonder whether influencing that guidance by sharing non-invasive test data, and potentially introducing an approvable surrogate in subpart H in the first instance might actually be useful. And in general, I'm not a massive fan of unapproved surrogates because they can get us into difficulty, but do wonder now whether in that population who really is at the greatest need of treatment and where there are still relatively few studies, that is somewhere where we could really influence the agency.
Well, I heard a very clear request to submit even just a simple slide deck to the docket. So again, where there are data, they should be reviewed by the regulator. That might be a really important first step right there. And then you make a great point that the time to endpoint for someone with advanced HIV disease and severe immunocompromise might be longer than someone per se with like F2 or F3 fibrosis and NASH. So maybe what we need to be thinking about are, do we have the right endpoints? Is mortality the appropriate or morbidity the right endpoint in that patient population? Are there other patient-reported outcomes, the feel part of feel function and survive, the function part of function that might be more relevant? I see Amy smiling here. Maybe she has something to add, because maybe instead of focusing on the biopsy and so much and how to make it better, which is important, don't get me wrong, maybe we can also be rethinking what are relevant clinical outcomes in a fibrotic NASH population that's non-cirrhotic.
Yeah, Erin, and appreciate all your comments, and Stephen, yours as well, and everyone else on the podcast. I hear all of this, and I think, what are we trying to cause? Getting back to the why it matters, what is it, and how are we going to do it? And what are we trying to cause? We're really trying to impact patients' lives. I like to put it in the function and feel because it resonates both as a physician that treated so many patients and as well as in the role today and working with all of you. So if that's the case, looking at what exactly we're trying to cause will be exceedingly important, especially when we collectively work with regulatory bodies and patient advocates, in the sense of really understanding as we are embarking on this work, right? This force, if you will. And a couple of years from now, we'll look back at this moment in time and say, this was the moment of change that really started driving us to be that force. When we think about the patient's perspective, they don't want to be told after years of being said, well, your LFTs are a little high, or you should lose weight, or you should be exercising more. And we really don't have great guidance of how to do that either, outside of what we know. Then they're told they have cirrhosis. Then they're told that their liver's in bad shape, or even worse, that they might need a transplant or palliative care. So how do we think about this from the patient perspective and what's really going to happen in clinical practice, in the sense of, are they going to really be told as patients that they need biopsies once, twice, maybe 3 times a year, depending on the therapeutic and what could come to market? That's not reality, right? So when I see everybody collectively nodding their heads, because, if I was in the shoes of a patient and someone told me that, I'd probably go, you know what? No thanks. So we collectively really want to focus on what are we trying to drive here and how are we going to do it? And I was, optimistic hearing that there is openness at the regulatory body perspective. And I'm hopeful that we will have a strategy, depending on what type of analyses and what type of data we're going to be able to produce, to make that happen. And it will take some thinking outside the box in order to make that happen. And we will need to collectively pull together to be able to provide that data in order to move the needle, because individually it's not going to be as effective if we all go and have data sets as individuals or siloed as opposed to a collective.
That all makes a ton of sense. I have 2 thoughts I want to add that are each a little bit off to the side, but still on this basic topic. Number one is I went back after chatting with a couple of people after Saturday night, and I went and looked at the podcast notes that we took on the January 31st meeting, which is to this day the second most listened episode in the history of the podcast, with what we talked about right after that. And when I contrast our reaction to what Joe Turner and Frank Anania had to say that day and the general reactions to what they were saying Saturday night, it's clear there's just been, movement within administrative government agencies could politely be described as glacial on a good day. But this looks a little more like you can see that the glacier has moved or melted or done something. It's not in the same place it was. Then they said, well, show us something. But now I think they're a lot more clear about what they want to see and where they need to see and where they know that they don't have good enough answers. To me, that's huge. And anytime a government agency says, we're willing to talk about what we don't know, as compared to, well, show us what we need to know, that's a big step in the direction. I'm also mindful of Terry Milton coming on this podcast as patient advocate after the patient-focused drug development meeting and mentioning that Joe Turner had texted at some point in the meeting that if he knew at the end of that meeting what he had learned several years earlier, there would have been different decisions made, which Donna, I think on the podcast, took to be about OCA. But whether it was about OCA or not, it was about an acknowledgement that goes, we've been doing this in a way that's not sensitive to key elements of the situation. So that's pivotal, number one, and collectively that left me with a pretty good feeling about the meeting. Number two, one of the things Stephen has been persuading me of over the last just couple, 3 weeks, but I think it's important, is that there's the result at the end, and then there's what do we do at the beginning in the context of screen fail rates and economics. We were talking about this yesterday, but it was only the 4 of us, so I'll share it with you guys and whoever else listens. When you talk about a screen fail rate dropping from 80% to 50%, if you flip those numbers, you're talking about an acceptance rate that goes from 20% to 50%, which means you are accepting 2.5 times as many patients. And that even feels bigger to me than 80 to 50. And in fact, it is bigger. And economically, it's way bigger. So to the degree that there's openness at the beginning to look at the role of NITs so that we can do that 80 to 50 thing, or even go lower than that, then that makes the economics of trials, the ability to develop data quickly, the willingness to take risks, all that stuff changes because the cost of doing it changes. And then if at the same time we're working on the back end, it feels to me kind of like a pincer movement. Stephen, I don't use a lot of military analogies, but I'll go with this one. It feels to me like a pincer movement. We're kind of flanking on both sides, at the beginning and at the end.
Let me just tag onto that, because I don't feel like it's gotten the justice that it needs to have, and that is anybody enrolling NASH trials right now, and you're looking at histopathology and you're incorporating MRI or some sort of mid-term assessment. In other words, to screen patients in, we go through labs, MRI, and biopsy. You're looking at screen fail rates of north of 80%. I don't care who you are, that is cost prohibitive. And in the setting of that, you also have incredible extensions of timelines to enroll patients into the study. So we need to be mindful of that. And if we're going to continue to generate interest from investors and pharma in the field of NASH, look, we still have a long way to go. We haven't even really touched the iceberg on combination therapies. So we need to be mindful of enriching patients for enrollment into these trials. And I've spent the past decade looking at pre-screen strategy to enhance enrollment. And we had a pretty good handle on it until we went and changed the paradigm on reading histopathology from a single pathologist to multiple pathologists. And we showed you the data on what that means relative to screen fail rates. It rises dramatically. So we have to get to a point where we can bring that screen fail rate back down. In the setting of having to look at histopathology, the provocative data that I think was presented here was do something simple like just look at more liver tissue. You're already collecting it. Why are you leaving most of it in a block? look at it and help that drive your inclusion criteria. And at the end of the day, you should see an effect on placebo response rate. I mean, I was struck when I was putting those slides together that you can look at the SEMA data and you can look at the lanifibranor data and you can look at the fibrosis endpoint, and the response to the high dose for both drugs was exactly the same. One hit stat sig and one didn't because of the placebo response. And that's exactly the same thing we saw with elafibranor and obeticholic acid. So there's a huge thing with this placebo response that's variable, and it's not necessarily driven by improving lifestyle in the placebo cohort. So that's another big part of this that I think is worthy of reflection on coming out of NASH-TAG.
Can I just ask, it's just quickly as something to Stephen was saying, there was a lot of talk last year about platform trials and combining the placebo arm to be able to use that. Is that where there could be a strength so that everybody is compared to the same placebo arm, if I understand platform trials better?
Look, it could be. The problem is there's just some issues relative to platform trials, probably more than we have time to get into here. But until we can have a better understanding of the role of nutrition in placebo response, the role of the natural history of the placebo, and then the role of the heterogeneity of the liver biopsy in placebo, it's hard to really understand what that's going to be and how a platform trial could potentially improve on that. I think the reality of the situation, Louise, is the liver is telling us to look at more tissue. And when you look at 3 H&E slides, you're looking at 60 centimeters of tissue or 60 millimeters of tissue rather than 20. You're looking at 3 times as much volume, and that's where you begin to pick up and smooth out some of the heterogeneity that we see across a biopsy sample.
But Stephen, even in light of that, isn't the fact that 9 hepatopathologists can't even agree on most bleeding hepatocytes saying maybe we shouldn't be spending as much time looking at the liver tissue in the first place? So
absolutely, we are. But I can tell you, the trials that are enrolling patients today, they don't have the luxury of shifting away in the middle of their trial to an NIT. So you have to deal with what you have to deal with. And right now, something that's simple and easy that could be done tomorrow at every company currently enrolling a trial is look at more tissue.
But if I can just add one point, because I really wanted to ask this question on the panel and I wasn't able to do so. And Louise, I'd love it if you would be able to respond to this. But, as we talk about reducing screen failure rates and making more patients eligible, I mean, there's also the patients that never get screened in the first place because the requirements of the trial are something that they're just not willing to do. So how generalizable are these data to the overall patient population anyway, if we're not necessarily using relevant entry criteria, or relevant endpoints? That's sort of a big picture aha moment that I took away from this, is that we're doing all of this work and spending all this money and all of this time, and then what do we have in the end? Is it something that the patients can even really look at and say, I really want to take this drug, or I really don't want to take this drug?
Okay, let's get a couple of responses to that, and then we are going to go to close because we're running into people's schedules. Louise, Erin specifically asked for your feedback on it, so why don't you go first, and then whoever else wants to jump in.
I think each person is individual. From my experience, there is always a trigger for somebody to make a change. Now, some of that could be that the medication option is the best way that they see for resolution and to be part of that. Everybody who participates in a clinical trial has an altruistic streak, and it's finding out what that is. There's a lot of people who, certainly when I've come across it, whose families didn't want them to take part in some clinical trials, but chose to do so. So it is very much individual. The one thing that I was struck by is something I saw a very long time ago, was a quality of life study. The people who get the best quality of life post-transplant are the people who go into transplant. The people who had the worst quality of life were the people who had an acute liver failure who woke up with the transplant, and we could never correct their quality of life because of the shock. So preparation is key to whatever we do. We have to be prepared. A sudden shock, we never get the right response. So the more we can find out about our population, the better our outcomes personally will be, because the better their engagement. Hopefully that answers something, but...
Any other follow-up thoughts to that?
So I've got a couple there. I guess the thrust of the paper that Stephen showed during his talk that we did looking at screen fail rates is around that, is about how applicable the results of a very tightly and highly selected population within the wider population. And we did a review a few years ago now looking at how comparable trial populations were with natural history cohorts and others, and they're quite different. The confidence that you would have necessarily that the findings would directly translate to those other groups is not necessarily particularly high. The relative reduction in progression to cirrhosis may be the same, but the actual rates of progression may be very different. So the overall absolute effect of treatment might be quite different. Talking about ballooning and saying it can be difficult to find, though nobody agrees with it, strikes right at the heart of saying, well, what is NASH? It is defined by ballooning degeneration. And if we're saying that we don't believe that, can't see it, it means that it's an opportunity either to say we can move away from it, but it strikes right at the heart of what we do and what we've been talking about for the last decade. Maybe the mistake was starting with liver biopsy and talking about change in NASH indicative of injury like we did for hepatitis B and C. But we're so far down the line now, it's very difficult to change course because of the studies that are running and in fairness to the sponsors who've invested up till now. The other thing that I would say about patients not being prepared to undergo liver biopsy in terms of treatment selection in the future, when we're defining non-invasive tests for use in trials, we must have an eye on the applicability for practice. There's a lot of enthusiasm about MRE, but I am not going to be able to MRE 15% of the adult population in Leeds. That is not going to work. So that we have to think about what the trial selection criteria will be and what the how you assess treatment response, so that we've got something that I can use every day in clinic. Ideally, a blood test or a bedside imaging biomarker. But MRE is not going to do that for me, and I think there are significant risks, a bit like liver biopsy, if we use MRE for widespread treatment adoption at the end of the day.
Excellent comments, and that's what I love about these type of meetings, even on a podcast, because what you hear is, here are some of the challenges, and let's explore those solutions. And so what I'm hearing is that we really want to better understand the disease, the course of the disease, progression of the disease. And one way to do that, besides what's already been suggested, is to really expand that patient population and really get that reach, right? And in the U.S., that can be a bit challenging, but I'm thinking, what about primary care? What about obstetrics and gynecology clinics? That's another area that is potentially untapped opportunity for us to explore that patient population, and that will help us better understand patient progression. The other comments that I wholeheartedly agree with is whatever collectively is going to be developed and will hopefully achieve the goal of reflecting the magnitude of biopsy so that we are able to see a change in how we conduct clinical trials is really an opportunity for us to better reflect clinical practice, because at the bottom line, the patients are going to be treated in clinics outside of clinical development and trials, and ultimately what are those tools and resources to not only diagnose and use as prognostics, but also for treatment management and treatment response, right? And the last thing we want is for patients to be given a diagnosis, given a treatment, and they're not responding. How do we monitor that, right, in the most optimal way? So I think there's a lot of opportunity for all of us collectively and some challenges that we still need to look at, but it's great discussion, and that's the first step. And then the how, right, of making that happen.
So I'm going to make a note, and then we'll go to close. The note is, I owe you guys, we owe our audience, and Amy, I'm going to lean on you on this one because that comment is right at the heart of it. An episode that goes at ways to enhance trial populations that are economically viable and clinically appropriate. Because one of the things I think we've all said today in different ways is more data faster will make lots of good things happen. We need to spend some time talking about how to do that. And this podcast is, I think, a good place to do that. And who else comes back? But I'm going to invite you to work with me on that if you're interested. Erin, you too, as two pharma people. Closing question, really simple. Same question I used yesterday. So Stephen and Louise, you can cheat. Try not to use the same answers. Something you hope to see that's different in a year as a result of this meeting, and something you think you will see in a year that's different as a result of this meeting. They can be the same thing if they have to. Brave one, go first.
I suppose from this, following on, I think it was Erin that mentioned it, maybe having patients around the forum next year or the next time we have NASH-TAG for the cosy fireside chats, because I think they'll be a permanent fixture, I have a sense, Stephen, as they've gone down so well.
Rachel, you've been nodding the whole meeting. You get to go next.
One thing that Stephen said during the meeting that really stuck out for me is that non-invasive tests are not used alone. So we would never bulk fibrosis in with NASH. But maybe the problem with developing new non-invasive tests is that we're bulking one or several tools for NASH itself. So I would be interested to see studies coming out that look at steatosis, hepatocyte ballooning, lobular inflammation separately as individual tests. So that's something that I'm interested in seeing. going forward?
I guess I can go. So my hope, and I think my belief too, is that a year from now, we'll be having another positive phase 3 NASH study result to discuss in detail at the meeting, which I think will be a huge advance forward. And that also advances in spaces outside of NASH will have more treatment options available for COVID and more prevention options available for COVID. So that'll make us all more comfortable about actually having even more in-person participation at next year's meeting.
So I'll go next. What I hope to see at next year's meeting is that we've actually taken steps to address these challenges and these opportunities, and we do have solid plans or actionables, deliverables, if you will, on these potential solutions. Because I think they're important, and I hope that we have the opportunity to explore them more robustly and then really find the step forward to have that data that's available so that we are able to move the needle, if you will, for all our patients with NASH.
Excellent. Thanks. Ian?
Yeah, I guess it goes back to seeing positive phase 3 data, which I expect that we will see. But I guess having had a discussion about NITs, that what I really want to see from that is consistent changes in non-invasive tests that correlate with histology and would give us confidence that that might go forward to improvements in outcome in the end.
I would like nothing more than to report on the first positive results that lead to FDA approval of a drug. I mean, just thinking back 6 years ago when I sat down and designed the protocol for the first THR beta, there was a lot of naysayers in the audience, and I would like nothing more than to show that that drug is effective in our patients with NASH. I think what I would like to see is something just more 30,000 foot level, and that is building on the consensus. I get the sense now that we're getting away from everybody doing their individual thing, trying to advance the field. And now there's a much more corporate effort to drive that we're coalescing into a union of people that are driving change. It's a little bit like 1776 again. We're actually, instead of just talking about it at different taverns, and you can imagine the people back in the mid 1700s, they were so disgruntled, with the British government oversight. And then at the end of the day, Ian, I hope I'm not throwing a stone your way, but at the end of the day, I mean, they rallied around and you see what happened. And I feel like it's almost a revolution in the field of NASH, and that movement has begun, and I would just like to see that continue. And when we have this discussion a year from now, we're going to look back and look at all the positive things that came about because of the corporate drive that we had collectively.
You do realise we've gone from Vlad's cult last year to now a revolution.
Actually, we've gone from Vlad on this podcast a year and a half ago saying, I only worry about the liver and I only worry about it in limited spaces, to Vlad's cult, to this revolution, and Vlad talking about cirrhosis. A lot of progress. I'm going to hit singles, and I'm going to go for small victories rather than large ones. I believe that within the next year, digital histopathology will have become a lot more important in this process and will have moved way up the field. What I'm hoping is that doesn't mean we will have integrated merely mean that we will have integrated what PathAI and HistoIndex are doing now, but we will appreciate that the greatness of artificial intelligence isn't when you ask it to emulate a human mind that is sloppy and inconsistent, but when you in fact ask everything that you can find in a data set that you didn't know existed in the first place. So my hope would be that within a year, we're in a place where we can take full advantage of the tools that offers. And I think for reasons we've talked about, we're going to be living with histopathology for a while. But if we can make it digital, and if we can make it not just about doing a better job of the things that humans are doing now, but about figuring out everything that's in those profiles, I think we'll be in a much, much, much better place in terms of being able to step forward. Anyone have anything else they want to add? We're a little bit over.
Well, I just will add that to have this big of an afternoon chat, not a fireside chat, but a post-fireside chat discussion, again, speaks to the interest corporately in trying to drive change. And look, we had 7 people on today. I would love to see another round of 7 people on, because I think we did a great job collectively of working through some of the issues that were discussed and even refining those a bit more, which is very helpful when I think about what needs to be done. It's great to bounce ideas off of great minds like yours, and then that allows me to be a little bit more clear in the direction that I personally would like to see this field going in. The more that we can drive that collectively, the better off we're going to be. I don't want to go off in a direction where I'm spinning my wheels in a particular aspect of this disease, but it's not really the focus that we really need to be going in. And this allows for that focus to really happen. It's an avenue that's enduring. I mean, every week we have this discussion rather than waiting 6 months for another meeting to get together and discuss it. So that's the beauty, I think, of the podcast.
So, Stephen, just to tell everyone else who's on this podcast and everyone who's listening to it, Stephen and I have texted a little bit during this podcast, but not about that. My close was going to be to say this shouldn't be the last time we do 7 people. That this has been really great experience. I mean, we've gone a lot wider than we usually do, but we've managed to go wide and reasonably deep at the same time. I think that's a real compliment to Amy and to Rachel and to Ian and to Erin, as not just because that's the order you are on my screen right now, not because any one of you did better or worse than the others. And we should think about Stephen and Louise doing this every 6 or 8 weeks, just bringing on 4 or 5 other people, picking a big topic and doing what we just did here, because I think this is really great. And I think it will be, as we go out to our audience, which is also diverse, it will give them a lot more things they can take out of this. So I want to thank the 4 of you for being pioneers on something that I think is going to turn out to be fantastically successful. And Stephen and Louise, as always, for everything. And with that, let's everybody say goodbye, and then I will turn it off and I'll come back with the business section in a couple of minutes. Bye-bye, everybody.
Thanks so much.
Thanks. It was great.
Bye.
It was wonderful. Thank you.
Welcome to today's business section.
This is our 3rd straight day of recording, and each session better than the last. To save my voice and get right to work on prepping this episode, I will limit my comments to a little about NASH-TAG and today's session, and then a lot of buzz and a little bit of vault.
Recording the NASH-TAG sessions took a lot of effort. We want to thank the special people who made this all possible.
I've made this point at the end of the last 2 episodes, but lots of people did some fairly heroic things to help this weekend happen. On the production end, thanks first to Mike Wilson, who had less than his usual 1-track per person sound to work with and still produced quality episodes. And thanks to the folks at Wasatch AV, particularly Zach, who came in on short notice, set us up with a creative solution that allowed us to record, and treated us like the most important customer in the world, something Mike does every week. Now, we weren't, but isn't that what everyone in customer service should do all the time? And beyond that, I'm blessed with Mike and with Eric Rounds, who do that every week and have been on that for at least a year and a half at this point. Also, I want to thank all our guests for the weekend and today. The weekend guests all stayed up very late if they're European, or started very early if they were north at the meeting, to maintain our demanding podcast schedule. So by name, I'd like to express deepest gratitude to Stephen, to Louise, Maza Nuruddin, Quentin Anstey, Scott Friedman, Donna Cryer, and Jorn Schottenberg for their weekend sessions. And in addition to the regular surfers, Amy Articolo, Erin Quirk, Ian Rowe, and Rachel Zayas for today's event. Unfortunately, all our surprises meant that we could not do live audience at NASH-TAG. Apologies to those who went looking for it and could not find it. We were back with live audience today, and I know there were a few people there, and we will be doing so in the future, and we will reintegrate the question section for next week.
Should we do this kind of session again? How can we do it better next time?
As I mentioned at the top of today's podcast, we've never had 7 people at once or 2 pharma people at once on the same podcast. Now, personally, I like the larger group and the more diffuse set of perspectives, particularly for a broad topic like NASH-TAG total. More importantly, I want to know how you feel. Please write us at questions@surfingnash.com or post a note on the LinkedIn discussion group or just my personal LinkedIn to tell us how you think this format worked and whether there are other big picture issues that would do well in this setup as well.
The buzz is buzzier than ever, and the vault remains wide open.
I provided some really striking listener stats last week. Here are a couple more. We had 13,000 downloads in the 4th quarter of 2021, which is roughly 1,000 per week, and roughly 85% more than we had had in any previous quarter before that. In the first 10 days of 2022, we've had 2,356 downloads as of 3:30 today, which would translate to something in the range of 1,700 a week. I don't think we're going to hold that all quarter, but it's a good start. And thanks to all of you who have listened and particularly gone back into the vault to find older episodes. By the way, we've had visits from 32 countries and downloads from the same number this week, which is bigger than the 25 we started last year at. We scored the #1 medical podcast in Bahrain earlier this week. That's the second time we've been #1 in the country, Macedonia being the last one a few months ago. Thanks all for downloading, listening, and sticking with us. In terms of the vault, this week's vault report starts once more with the Paris NASH imaging conversation 46.2, which gained another 71 new downloads in the last week to go to 1,194 as of 4 o'clock Eastern Time on Monday. Conversation 57.1 for the AASLD wrap-up episode also continues to do well. In fact, it's now at 256 downloads and number 4 on the all-time list, up from 7 last week. Beyond that, the full episodes 61 about the Splendor study and 62 and 63 as first parts of the year-end wrap-up have done really well. Among earlier year 2 conversations, 30.2 2, which focused on the importance of common, simple, reliable NITs in the context of Global Liver Institute's U.S. NASH Action Plan, grew more than 5% in the week. So with that, let me just wrap up. I want to thank our team, the Magic Mike Wilson, who's been pressed even beyond his normal duty this week. Although that happens so much, it almost doesn't make sense for me to say it anymore. There's a new normal on this. The creative and highly energizing Eric Rounds, and the pragmatic and strategic Steve Erin, whose work we are actually heading to start doing now. Most of all, I want to thank you, the listeners, who provide energy, inspiration for all of us to do this every week, sometimes 3 days in a row, sometimes early in the morning or finishing late at night. Listen, Omicron is putting a dent in all of our lives, including possibly mine. So all I can do for today is I can wish all of you good health and forbearance as we go through one more round of this COVID mess. Stay safe, mask up, practice social distancing, surf on. I look forward to seeing all of you next week. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com and we will answer on the podcast or the website.